Hello, everyone. Thank you so much to join my talk, and it is really my honor to have this opportunity to join this CRISPR virtual event and share some of our recent studies in terms of using CRISPR-Cas12 systems to build up our autocatalysis reactions for biosensor development. So when we talk about the CRISPR-Cas systems, now we understand it represents a huge protein families including Cas9s, Cas12as, 30s, 40s, etc. And all of these CRISPR-Cas proteins has unique functions to be guided by a short piece of RNA, or the guide RNAs, to find and bond to its complementary sequence and activate the Cas protein's endonuclease functions to specifically cutting its target sequences. And this kind of unique phenomenon has been developed into a novel biotechnology toolbox and has been applied in all sorts of applications, especially in the gene editing functions, which has been recognized as the magic gene clippers. Well, in the past few years, there also has a very unique transcleavage function has been discovered in type 5 and type 6 CRISPR-Cas systems. And when we talk about these unique transcleavages, let's take Cas12a as examples. The Cas12a will bond to its guide RNAs and is capable of finding bound its target sequences. And after it bound to its target sequences, the Cas12a will be activated and firstly specifically cutting its complementary sequences, which has been called as a 6 cleavages. But after these specific cuttings, activated CRISPR-Cas12a will continuously cutting its surrounding single-strand DNAs in a random format. So we can say at these circumstances, if we are introduced a single-strand DNA-based probes or reporters, the continuous degrading of the single-strand DNA will cause signal amplification procedures that we can be detected in the downstreams for biosensor development. And this kind of unique discovery has pushed the development of CRISPR-Cas-based biosensors, especially in the past 5 years, which has been developed in all sorts of different biosensing systems, and which we can also be verified in the rapid increased publication numbers in regarding to the CRISPR-Cas diagnosis and the detections. And currently, with the development of CRISPR-Cas biosensor, we noticed that it has a unique or all sorts of different superior biosensing performance, such as it can reach even single nucleotide differentiation capabilities. And also, the sensitivity can reach to a picomolar ranges without any additional applications for a standard CRISPR-Cas transcleavage reactions. And when we combine with amplification studies, it can reach even to attomolar to zmolar sensitivities, and the overall reaction can be run at the isothermal conditions under 37 degrees. And the overall transcleavage reaction can be done by within half hours. And now CRISPR-Cas biosensor has been extensively applied in all sorts of biosensor areas. Because of its excellent versatilities, it has been able to combine with different type of other biosensor technologies, such as isothermal applications with electrochemical chips, and also combining with immunoassays and adding to other kind of microfluidic devices, and capable of differentiate or detect of all sorts of analytes, including DNAs, RNAs, proteins, and also small molecules. However, despite all the great achievement CRISPR-Cas has been able to achieve, there still have some limitations, especially when we are talking about applying to, like, let's say, clinical or environmental diagnostic scenarios, which usually requires PCR level sensitivities or we can see 1 copy per mils or 1 automolar sensitivities. Although, as we just said, CRISPR-Cas biosensing can reach these sensitivities, but normally it requires additional or external amplification technologies, such as the use of RPA lamps or use of uniquely or customized developed devices. But by using or adding all these other technologies, it also increases the overall system complexities and also limits the reliabilities and make the reproducibility sometimes can be challenges. And also it can also increase the longer reaction times and get the cost a little bit higher. And most importantly, when we combine with application, like especially nucleus-based application strategies, there has the potential of causing amplicon contaminations, which can lead to a false positive in downstream detections. So we were thinking this kind of limitation in the key point is because the limit of 1 target can only activate 1 CRISPR-Cas RNPs and then generate the final signal for detections. We're thinking, is there any possibilities that we can make the CRISPR-Cas systems activated by a single target, and then the activated Cas12a RNPs will be able to amplify such activation scenarios to eventually harness a whole bunch of activated Cas12as to generate extensively amplified signals for detections. So under these circumstances, we will be able to make all reactions in a single port at room temperatures, and no need to add in other complex reactions except the standard CRISPR-Cas reagents, and no need for additional applications, but still reach the requirements for clinical detections. So in our recent studies, interestingly, we found that the target DNA structures can actually affect the Cas12a's RNP activation efficiencies. So by here, we firstly circled a small piece of linear DNAs into a circularized structures by using either a T4-like method or a clean chemistry-like methods. And then we find out, compared to the linear formation of the single-strand DNAs, the circularized single-strand DNA will be able to considerably or significantly suppress the activation of Cas12a RNPs. And we suspect it's because these such small circular single-strand DNA structures can actually cause topological barriers for the Cas12 RNP to recognize and bind to its target regions. And by building this fund, we are thinking about further develop this or design these small circular DNA nanostructures to better control the Cas12 activations. So here, in addition to the circular single-strand DNAs, we was adding another layer of shorter complementary DNA, or the cDNAs, to form a partially double-strand DNA structure here, which combines the overall circular DNA nanostructure will combine 3 parts. One is the pan regions, one is the blue regions, which will be able to bound, which has the complementary sequence to the guiding RNAs, and also a small black region is maintained as a single-strand DNA structures. And this kind of circular DNA nanostructure, as we tested, compared to its linear formations, it can also suppress its Cas12a activation functions, although it's slightly higher than the negative control. But compared to the linear DNA format, it has significantly reduced fluorescent signals after the activations. And more importantly, if we are putting these this circular DNA nanostructure into a pre-activated CRISPR-Cas reactions, this small black region will be able to cut by the transcleavage of previously activated Cas12a RNPs, then put it back to the linear formation. And our data shows after the relinearized formations, this kind of circular DNA structure go back to a linear formation, and it can also be used to activate another round of CRISPR-Cas transcleavage reactions. And build on this, we can say the linear formation before circularize, it can activate the CRISPR-Cas. But when we circularize it, its activation has been restricted. But when this small black region has been cut by the pre-activated CRISPR-Cas RNPs, the relinearized one can also be used to activate CRISPR-Cas12a. And most importantly, we also identified the length or the size of the circular DNA structure play a key role in control the Cas12a activation here. So when we try to get the circular length shorter and shorter, the activation Cas12a transcleavage dropped significantly. But there has also another downside is if we shorten it too much, even the linear formation will not be able to activate the Cas12a. That's what we don't want to say. But on the other hand, if we get the circle bigger by elongate the linear sequence, the capability to suppress the Cas12a activation also be reduced. So the size is really important to choose in a very short regions. And by doing this, eventually we will be able to use this circularized DNA nanostructure to establish autocatalysis schemes by using the Cas12a transcleavages. Basically, if we have one of the CRISPR-Cas12a RNP being activated by the actual target's DNA, the activated CRISPR-Cas RNP will be able to cut the small single-strand regions on the circular DNA nanostructures and put this circular DNA structure back to linear format. And the linear format DNA nanostructure will go back to activate more Cas12a RNPs with the guiding RNA complementary to itself sequence, and then to cut more of the surrounding single circular DNA nanostructures and eventually close this autocatalysis loop. So build on this, our results actually show that by applying this circular DNA nanostructure, or we call it the cell mediators here, the overall reaction intensity response to the same concentration of targets has been dramatically increased, as here can show compared to the normal CRISPR-Cas or the standard CRISPR-Cas reactions, the cell-mediated added reaction has more than 7 times higher signal increase for 3,060 seconds. And after 2 hours, it even has more than 14 times fluorescent signal increase. And more importantly, when we compare the transcleavage patterns, the circular mediated added CRISPR-Cas reactions can actually transfer the previously linear pattern of the transcleavage for a standard CRISPR-Cas to an exponential signal generation patterns. And we can also identify that with the increase of the circular mediator or the circular DNA nanostructures concentration into the reactions, the overall signal amplification schemes has been continually increased. As we can see here, if we increase the cell mediator concentration from 5 nanomolars to 25 nanomolars, we can see a huge increase in the final concentration, in the final fluorescent signals, which has been released by the transcleavages. And by doing this, we actually changed the pattern of activation targets to CRISPR-Cas12 activations. And originally, as previously we understand, normally one target nucleic acid can activate one Cas12a RNPs at a certain times. But here, with the circular DNA long structure we applied, one target nucleic acid can activate even more than 1,000 of CRISPR-Cas RNPs in a single port reactions. And we also find that there has linear correlations between how many Cas12a RNPs can be activated in response to a single target nucleic acid is positively correlated to the concentration of circular mediator has been added to the reaction systems. So to now, we actually realized our aims to break the barrier of one-to-one Cas12a activation patterns response to a target with a multiple CRISPR-Cas RNP, activate CRISPR RNPs responsive to the presence of a single nuclear targets. And after this, we were thinking about, we are introduced a circular DNA nanostructure into a standard CRISPR-Cas reactions, but there still has a circular DNA added and also a reporter to generate the final signals. So we're thinking, is there any possibility we can make the system even simpler, just as what we have using for a very basic standard CRISPR-Cas12a reactions? So here we try to combine our circular DNA structures with the normal or the previously commonly used single-strand DNA formed reporters. So here we put the fluorophore and the quenchers to each side of the cDNAs on our circular DNA nanostructures and form this bifunctional DNA nanostructure, with which on one hand, it can still drive the autocatalysis reactions as previously we just talked about, but also act as efficient reporters to actually produce the amplified fluorescent signals for detections. So by combining these two together, we have created exactly the same assay procedures as a standard CRISPR synthesis, only has CRISPR-Cas12a RNPs and a reporter in the reaction solutions. But the difference is these circular reporters can induce the autocatalysis reactions to make the overall system response to the presence of a single nuclear target with multiple Cas12a RNP activation. And with this use of autocatalysis reactions, we finally will be able to build up a new autocatalysis biosensors with a very simple detection schemes, just as we normally use for the standard CRISPR-Cas reactions. Basically, we add the sample DNA and also maybe RNA, I will talk about it later, to the CRISPR-Cas autocatalysis reaction mixtures. And then we wait for 15 minutes at room temperature. Then go to the device to test the signals for the final detections. And the overall detection can be controlled even within 15 minutes at a single spot. And the whole reaction has no complex reagents being added, only the circular DNA, which already formed as reporters. And there is no additional amplifications in a single pore reactions under room temperatures. But still, as we can see, compared with the classic or standard CRISPR-Cas transcleavage reactions, which will not be able to respond to the concentration changes of targets lower than 100 attomolars, our cell mediator-based CRISPR-Cas12a autocatalytic biosensor can respond to even the presence of 1 attomolar targets, which can meet the clinical standards for nuclease detections. So then we start to apply this kind of Cas12a autocatalytic biosensors to all sorts of detections. Firstly, we try to detect the helicobacterial bacterial DNAs. As we can see here, we will be able to reach maximally 1 attomolar sensitivities and with 3 logs of linear ranges from 1 attomolar to 100 attomolars. And we're also trying to apply to actual clinical sample detections for the HP genomic DNAs. Here we tried to detect the HP genomic DNA in sparked saliva samples. And we will be able to maximally differentiate the presence of around 1.4 copies per mil HP genomic DNAs from the samples. And then we also, because we know CRISPR-Cas can respond to RNAs directly, as reference has been demonstrated and we also tested. But normally, CRISPR-Cas cannot directly used for RNA detections because the RNA-triggered Cas12a transcleavage, the intensity is extremely low, much lower than compared to DNA targets. But because the autocatalysis reaction in the second round can put the overall activation patterns back to DNA activations. So our system can also be applied to RNA detections. And we tested here by using the SARS-CoV-2 viral RNAs. And we can also get the system response to the presence of maximally 1 attomolar viral RNAs, and also with 3 logs of linear ranges from 1 attomolar to 1,000 attomolars. And we also try to use sparked clinical samples to test the capability of differential genomic RNAs, genomic viral RNAs from the samples. And you can see here, we will be able to differentiate the presence of even lower than 1 copy per mil viral RNAs from the sparked samples. And after this, we also try to apply our autocatalysis detection systems to detect the circulating tumor or ctDNAs. As you can see here in figure A, we tried the systems both using the cell mediator plus reporter system, and also the one-case RMP system, which is the combined cell mediator and reporter systems. And both these two autocatalysis reactions can generate much higher fluorescent signals compared to the standard CRISPR-Cas reactions in terms of response to the presence of ctDNAs. And we'll also be able to differentiate maximally even 1 attomolar ctDNAs from the samples, either from the PBS samples or the Spark in plasmid samples. And after that, we're trying to apply these systems to animal studies by using the colorectal cancer mouse. And we took the colorectal cancer mouse plasmid and directly use a plasmid for the ctDNA detections using our autocatalysis reaction systems. And we can see here, instead of response nothing by using a normal CRISPR-Cas reactions, our autocatalysis system can actually respond to the concentration change of the target ctDNAs in the animal plasmids. As you can see here, with the colorectal cancer happens, we can see the increase of ctDNA here. And after the treatment, after the X-ray treatment has been given, we can also identify the treatment response by seeing the slightly reduced ctDNA concentration in the plasmid. After we have done all of this tests, we also think about as normal CRISPR-Cas systems or the other common CRISPR-Cas systems can be easily combined with the lateral flow assays for naked eye detections. So I think is there possible for our system to also be able to apply it to lateral flow assays? So we try to modify our circular mediators to this way, to a circular amplifier we call here, which we replaced the original labeled fluorophore and quenchers by one is the biotin labeled in one side of the cDNA, and also elongate single-strand DNA at the other side with a fan fluorophores on it. By this slight modification of these circular DNA nanostructures, we will be able to put the reaction product directly onto a pre-made lateral flow strip, as any other reported CRISPR-Cas system have done, and be able to differentiate here the ctDNA concentration changes by naked eyes. So when we compare our circular mediator-based CRISPR-Cas12a autocatalysis biosensors to other type of application strategies used to combine with CRISPR-Cas reactions, our circular mediator can provide even faster reaction times to reduce the overall detections within half hours. And also, when we try to detect the RNAs, there's no need for us to use reverse transcriptions in the reaction systems. So it's a much easier single-pole reactions, which can be done even at room temperatures. And most importantly, without the generation of amplicons, which is much easier for us to implement these systems to different type of point-of-care detections, where it has much less environmental controls to deal with amplicon contamination issues. But we still need to be aware that this kind of systems also have its own limitations for current status. One is the difficulty to make a perfectly or the well-functional circular DNA nanostructures, because this DNA nanostructure is way too small. Synthesis of this structure is much harder than we previously think, especially if we want to scale it up. And also, we believe with further optimization of our systems in details, the performance, especially for the sensitivity and also the reaction times, can even be further improved, which will be better benefit for combining with other kind of different devices. So to sum up, now we understand that by manipulating the DNA nanostructures, we will be able to affect the Cas12a activation efficiencies or to affect the transcleavage efficiencies. And by using these specifically designed DNA nanostructures, we will be able to break through the previous barriers of one-to-one correspondence between the activated Cas12a RNPs per target nucleic acids. And because this unique structure of half double-strand DNA, half single-strand DNA structures, we will be able to use it to either control the activation or block of the Cas12a RNP functions. And by building the autocatalysis reactions using these small circular DNA structures, we will be able to reach 1 attomolar sensitivities at room temperature condition within 15 minutes, using pretty much a standard CRISPR-Cas12a reaction protocols. And there's no need for additional reagents, reactions, or devices. So it will be very easy for the future implementation to all sorts of other type of technology or devices, just as currently the standard CRISPR-Cas system has been done for biosensor applications. So before I finish my talk today, I would really like to thank first my supervisor, Professor Eva Godis in University of New South Wales, and also two of my colleagues, Dr. Fei Deng and Dr. Biyao Yang. And without their help and support, there's no way we can actually realize these systems we just talked about. And if anyone have any questions, please feel free to contact me at any time you want. And thank you very much.